Hello guys, I am Harun and it's your YouTube channel Harun Bayojon. Today in this video I'll discuss about oval hypothesis. This is very easy but sometimes it is complicated to someone. To explain this hypothesis very clearly and remove your any doubt about oval hypothesis. So what is oval hypothesis? According to Watson and Crick, adenine bind with thymine or uracil by 2 hydrogen bond and guanine bind with cytosine by 3 hydrogen bond. But in genetics, in molecular biology, there are 61 codon, 3 are nonsense codon. 61 codon represent 20 amino acid, okay, amino acid. That means one amino acid represented by many codons, okay. Example, leucine many codons. This is called degeneration of codon. In the other way, that for 61 codon only represent by less than 40 tRNA. Clear? It is not possible, okay, to produce several type of proteins or RNA require more tRNA. So this problem will be solved by oval hypothesis. This problem is solved by oval hypothesis. This hypothesis proposed by Francis Crick in 1966. According to this hypothesis, the 2 code of 2 nitrogen base of the mRNA, okay, 2 nitrogen base of codon, that is 1 and 2, are normally combined with the anticodon, okay. But in case of third base of codon, it can combine the third position of the codon anticodon bonding is unstable. The first and second position is stable by Watson-Crick model, okay, but the third position is unstable, okay. The third position do not maintain Watson and Crick rule, because here one anticodon nitrogen base combined with more than one codon nitrogen base, okay. Here in third position, the one anticodon nitrogen base can combine more than one codon nitrogen base. Say for example, in case of leucine, that is, suppose this is mRNA. Here the nitrogen base is CUC. Clear? The anticodon is GAC. Clear? GAC. CUC. So here the anticodon is CUG. So this anticodon is stable, but here G can combine with C, or G can combine with A, or G can combine with U. Clear? So here the one anticodon represent represent more than one codon. So the codon anticodon problem is solved, because 61 codon represented by less than 40 tRNA or anticodon. So if one anticodon determine or recognize more than one codon, so the problem is solved. So there is no problem in translation. Okay. There you can, I think you understand this. According to the overall hypothesis, only the first two base of the codon have precise pairing with the base of the anticodon tRNA. Okay. So there the two nitrogen base of codon is normally binding with two nitrogen base of the codon and anticodon by Orson and Crick rules. But while the pairing between the third base of the codon and anticodon may be oval, so this third base of codon and anticodon may be oval position. Okay, here the single anticodon can determine more than one codon nitrogen base. Clear? So less tRNA can easily represent 61 codons. Okay. The next point, here you can see very clearly. Suppose this is the third position. This is the third position of codon, and this is the first position of anticodon. If in the first position of anticodon guanine is present, it can bind with normal bind cytosine, but also bind with uracil. So guanine in anticodon region present guanine, it can bind with cytosine or uracil. Okay. If the anticodon region C is present, it can bind with guanine, normal, Orson and Crick model, and adenine bind with uracil, it is also normal. But in case of uracil, also it bind with adenine or guanine. Or if hypoxanthine is present, it can combine with uracil or cytosine or adenine. So one anticodon recognize more than one the codons. So there is no problem in less number of tRNA than codons. This problem is solved. So what is the significance? So less number of tRNA represent 61 codons easily. Clear? Next point is the oval allowing faster dissociation. That means when the stop codon appears, it easily or more fastly dissociate due to translation termination step. When the stop codon appears in the mRNA region, it can easily dissociate or fastly dissociate than Orson Crick model. Okay. The existence of oval minimize the damage that can be caused by misreading of the code. Suppose, say for example, here GAG present. If in case of C there is uracil, no problem, same amino acid. In case of A, no problem, or also leucine. In case of U, A or C, if the codon base is changed, C, U or A, third position, there is no nonsense codon or point mutation occur here. Okay? यहाँ पे कोई point mutation नहीं आएगा, क्योंकि this single anticodon nitrogen base can combine is more than on codon nitrogen base. So if G Combine with normal C, no problem. Combine with U, no problem. Combine with it, because 3 represent the same amino acid. Here some questions are present. Oval hypothesis deals with phenomenon of multiple codon coding for the code of single amino acid. Yes, single amino acid represented by more than one codon. A is right option. 2. A oval hypothesis explain the phenomenon of multiple codons coding for the code for a single amino acid. Multiple codon for single amino acid, right? On tRNA, molecule can recognize and bind more than one codon. Definitely, single tRNA can bind or can recognize more than one codon due to only this 3 position. Okay, this is also right. So B is the correct option. Both assertion and region are correct, but region is not the correct explanation of assertion. Clear? Next question. Which of the following represent the oval CuC encode deep or not? This is leucine. This code represent leucine. CuC encode for the same, also definitely leucine. The oval hypothesis is the occurrence of more than one code of amino acid. Single for single amino acid, there are more than one code. The example CuC, CuA, CuG, CuU, all are leucine. So for one single amino acid, more than one codon is present. I think you can understand the oval hypothesis. So thank you for watching this video.